This Interact educational resource, called Secrets of Dead Plants, explores how we can use herbarium specimens, that is, plants collected for identification purposes, for exploring past environments and environmental events. The resource is produced by Tom State University for Interact, Wicked Weather Watch, University of the Arctic and EduArctic.
This herbarium belongs to Tomsk State University. Professor Porfiry Nikitich Krylov has founded this herbarium at 1888. Plants were collected from the field and dried in order to preserve them so that we could identify species and describe new ones. Some of plants were collected recently, but some of them were collected centuries ago. Some of specimens give us a lot of information, much more than just their identity. One of example of these plants are LYCOPODIUM, ANNOTINUM, and other members of this ancient group of plants.
LYCOPODIUM ANNOTINUM is a club moss. It is a member of a very ancient group of plants called the lycopodids that lived several hundred millions of years ago. Today, it is not found as trees, but is found as low herbs. It's very common in the Arctic and particularly the subarctic. It does not reproduce by seeds, but reproduces by spores that come off as a cloud of dust from the reproductive structures called sporangia. The spores are so small that they can travel all around the world several times. And during their passage through the atmosphere, until they eventually fall on the ground and form a new plant, they experience the sun's rays and particularly ultraviolet B radiation. The three different types of radiation are absorbed differently when they hit the atmosphere, and they have different effects. UVB is one of the many types of energy that is emitted from the sun and travels through space, and some of it hits the Earth's atmosphere. There are three types of UV. One is ultraviolet A, the other is ultraviolet B, and the third is ultraviolet C. The Earth's atmosphere is a mixture of gases in a very thin layer surrounding the Earth, without which life would be impossible. Throughout the layers of the atmosphere, there is ozone, the chemical ozone, 3 molecules of oxygen, and they concentrate in the stratosphere between about 10 and 50 kilometers altitude. When the different types of radiation come through the stratosphere, some is totally absorbed, and UVB is also absorbed to some extent, even though some reaches the ground surface. However, you all know that there has been an ozone hole. It's not a complete hole, but it's where ozone is present in very low concentrations, and that allows more UVB to pass through into the atmosphere and eventually to the Earth's surface. The UVB radiation also contacts the spores of Lycopodium and, of course, many other seeds and spores in the atmosphere. But species that are protected against UVB radiation have chemicals in their cell walls, such as fumaric acid and ferulic acid, and these chemicals absorb most of the UVB, and by doing this, they prevent the UVB from reaching the nucleus of the cells, which have DNA, because the DNA is very sensitive and is easily damaged by UVB, causing, for example, mutations. So the chemicals in the outer layers of the spore wall protect the DNAs and reduce the mutations, many of which can be harmful. Some plants, and even parts of plants, like the spores of Lycopodium, are adapted to UVB radiation. So the more radiation that they experience, the more chemicals are produced to protect the DNA from harm. The graphic shows how the concentration of UV absorbing chemicals increases as the dose of UVB increases. We now have technology that is so sophisticated that we can separate spore walls, minute microscopic spore walls, from the spores and measure the chemicals in the spore walls. Now let's return to the dead plants. By measuring the amount of UVB absorbing chemicals in plants collected many years ago, we can actually back predict what the UVB was that they were experiencing, and therefore look at the ozone thickness in the atmosphere way back in time, before we had machines that could do that. Why is this important? Well, we know that a few hundred million years ago there was massive volcanic activity throughout the world, and particularly in Siberia, and this destroyed the ozone layer at the time. The resulting destruction of the ozone layer caused high UVB radiation to damage many plants and perhaps animals too, and many species became extinct. By finding fossil spores of the Lycopodium species, or the lycopsids, we can find mutated spores, and these undoubtedly mutated because they were receiving too much UVB because of the ozone hole. Knowing that problem in the past, we were very keen to prevent even smaller problems arising in our current time through our man-made impact on the ozone layer. In 1985, scientists found an ozone hole over the Antarctic. This is the blue area you see in the middle graph. This wasn't an actual hole, but it was rather a severe thinning of the ozone, particularly in the stratosphere, that allowed UVB to penetrate in greater concentrations to the Earth's surface. Scientists around the world then started to look at how the stratospheric ozone layer was being depleted by man-made substances. These are chlorofluorocarbons, CFCs, and halons, CFCs. These compounds are released in many areas of modern technology and had a severe effect on the stratospheric ozone layer. After the Antarctic ozone hole was found, then an ozone thinning was also found over the Arctic, and scientists began to look at what effect the increased UVB was having on people and the plants of these areas. The effects on people, for example, include eye cataracts and some skin cancers. The effects on plants, reduced growth, and so on. This event led to the formation of the Montreal Protocol, signed by most of the countries of the world, which limited ozone-depleting substances. Today, there are far fewer damaging substances released into the atmosphere as a result of the Montreal Protocol. We don't notice the change, but in fact in our aerosol sprays and our fridges and refrigerators and air conditioning units, the damaging substances have been replaced by chemicals that are far kinder to the stratosphere and thereby protect the ozone layer. What are the lessons we have learned by looking at the Lycopodium spores and our past UVB levels, and also into the distant past of hundreds of millions of years ago? The first lesson is we should be careful about the man-made substances we release into the environment, as we do not know always what effect they have. Secondly, the countries of the world have shown through the Montreal Protocol that they can work together to solve large environmental problems. And thirdly, the young generation should learn from the mistakes of older generations and use new knowledge and technical abilities to protect themselves by caring about their environment. This Interact educational resource has been developed by Vyacheslav Rudchenko of Tom State University and designed by Professor Terry Callaghan on behalf of Interact, the University of the Arctic, Wicked Weather Watch and EduArctic. The study has been based on a paper produced by Lomax and others, which was published in Nature Geosciences in 2008.